So back to the question of population regulation. Given that the population of insects doesn't go to infinity and doesn't necessarily drop off to zero, this idea of regulation, again, postulates that there's some boundedness and that the population fluctuates between. And when there is a deviation away from these bounds, there are some factors that pull the population back to occur within those bounds. And in order to do this, in order to have this return tendency, there needed to be some factors that, as the population got too big, pulled the population back down. And we modeled that in the one population discrete time equation by creating those differences in the growth rate parameter that varied as a function of density. And so people ask the question, does a system actually have to be regulated in order to be stable or persist? That is, does there have to be some of these factors that pull the population back in order for us to observe populations of organisms in nature? So one school of thought was that there are these density-dependent factors that act as regulators that pull populations back to an equilibrium. And in fact, the only way in which a population is regulated is by some kind of feedback process. And this feedback process, again, is reactive to some density of the organisms. This was a school of thought that was born in the 1930s that is often attributed to A.J. Nicholson in Australia. Sometimes this is referred to as the functional school. Varley and others in the UK kind of came up with similar ideas. And their concept was that competition or predation were those factors that can cause density dependence. As populations get too big, there's a resource limitation, which means there's starvation, and starvation is that thing which causes the populations to go back. That's competition. Or as populations get big enough, that you get predation of those organisms, and the predators is actually what brings the populations down. So how do you study density dependence? I'll give you a couple of examples. There's lots of different ways, but I'll give you a couple of examples. One is by actually following the populations very carefully and watching how they change as they move from one stage to the next and from year to year as well. And then seeing if those changes are somehow associated with density, and if there are factors that are associated with that density as well, like predators, for example. And I won't go into the detail of how to actually calculate these analyses here, but there's this kind of common classical way that's called a K-factor analysis that basically says compare your population at the beginning of a time step compared to what it is at the end of the time step, and calculate either a ratio or a fraction or something like that, and then compare that change, basically that delta between these two time steps, so like going from egg to larvae, compare that over time. This is the so-called observational approach. Go out into nature, measure these things, and see what happens. The experimental approach is actually to manipulate the populations or manipulate predators or other things and actually see what the consequences of. So this is a study that was kind of a classic study of the winter oak moth by Varley and others that occurred over many decades, actually. The winter oak moth actually has an interesting behavior where they actually come out as adults in the winter where there's no leaves. They mate, males. The females are wingless, so they kind of scoot around on the barks of the trees. They'll mate with the males. They lay eggs, and then as the buds begin to burst from as the leaves come out of bud in the spring, the eggs will hatch, and the caterpillars, which are these little geometrids here, will begin to munch on the leaves like that. And this beautiful illustration here shows kind of the life cycle. After they feed sufficiently on the leaves, they fall to the ground to pupate, and the researchers here put these little boxes that actually allowed them to collect the caterpillars as they moved to the ground. And then they capped the box here and watched what came out. And sometimes what you had coming out, instead of another moth, was a series of parasitoids, like these little wasps here. Some of them attack the moths relatively early here in June and July. Others kind of emerged a little bit later here in August and September. They emerged, they immediately reparasitize the larvae, and then would emerge the next spring. They're univoltine. They have a single generation per year. There's also another parasitoid, which is a fly, which actually emerges early on in the spring, and actually attacks the caterpillars themselves, and the caterpillars kind of bring it with them underground where they emerge, where they are parasitized, and then after they pupate, the parasitic fly actually emerges from them. So they can measure and keep track, basically, of the populations of these moths in very discrete ways over time and through their different instars. And what they were able to do is actually calculate the amount of mortality, this K value, the ratio of how many were there at the beginning to how many were there at the end, and see how it fluctuated over time. And this is the overall winter moth population here. You can see that this is on a log basis, so there's around 200 individuals per meter squared, and there's those little boxes. In other years, there's almost, that's 10 to the second, so that's 100 individuals per meter squared. Compared to other years, there's only 10 individuals per meter squared, 10 to the first. So these are log scales. So big fluctuations in densities over time. And the mortality of those individuals also fluctuates over time. And as you can see, there seems to be kind of a set proportion of individuals that are dying each year, pretty constant from year to year. When you break down those factors that are causing that mortality, you can see a different kind of a different set of patterns. Here, for example, is winter disappearance. It's just the larvae go in, they pupate, and then nothing ever comes out, and they can't tell what actually killed them. Maybe they dried out. Maybe who knows? They weren't measuring closely enough. So this is the overall mortality here. This is the overall general K factor, and this was that's attributed to winter disappearance. And the one that most closely resembles the overall mortality is often referred to as the key factor. This is the one that mostly sets the overall abundance of the organisms. It most closely resembles the overall mortality and is the key determinant of how big these populations are. You can see that there's a much smaller proportion of individuals that are parasitized and killed by the parasitic fly, parasitism by other insects, there's some pupal predators, and so on. So you can break down the various sources of mortality. Now, when you compare these K factors, the mortality caused by these different agents, to the density of the moths in that particular year, that will tell you something about whether mortality caused by predators or parasites or things like that is a function of density. So let's see what they found when they did that. So here is the abundance of the moths, and here's the amount of mortality caused by these different agents. Now, what's interesting is that the K factor, the overall, the factor that causes the most amount of mortality, winter disappearance, doesn't seem to be dependent on density. It seems to be there's no line here, and this relationship seems to be kind of a shotgun of points like this, suggesting that as the population gets bigger, the intensity of mortality, the fraction that is killed, doesn't really change. And so if winter mortality or winter disappearance is one of those feedback mechanisms that brings the population back as it starts getting too big, this isn't it. There's no density dependence in it. So it's high across this whole range of densities here. On the other hand, pupal predation by these beetles here, this little carabid and this rove beetle here, the staphylinid, actually is density dependent. So again, along here is density, and this is the degree of mortality caused by this particular factor. So as the density gets bigger and bigger, the fraction of individuals that are killed by these pupal predators actually increases. It's relatively low here, and it almost doubles here. And again, this is on a log scale, so this is actually a big effect. And most of these other factors, again, are either density independent, or actually in the case of these larval parasites, might even be negative density dependent. That is to say, as the population gets bigger, there's less parasitism by these wasps. So that's actually not a good thing as far as controlling populations. That means more and more of them are actually escaping parasitism if it's negative density dependence. So in this situation here, the authors conclude that, in fact, the population does have some factors that are density dependent. Well, what they found here is that, again, with this pupal predation, if you map this out over time, what you find is it's a very special kind of density dependence. They found that this kind of counterclockwise pattern here, where population in 1 year population size in 1 year actually causes greater mortality in the year after. So the predators might build up, and then they have a greater mortality effect on the subsequent generation. And you find these counterclockwise spiraling, which is evidence of delayed density dependence. So it's not density dependence in response to the population size that year that matters, but it's the density in the previous year that determines how intense that parasitism is going to be. So this is an example where, in fact, there is density dependence. It actually has a temporal effect, and it's due to a particular agent and not all of the agents acting on the winter moth mortality. So, okay, this is an interesting story. Is it unusual? Is this the norm with most insects, where populations are kind of ratcheted back by these density-dependent factors? There were a couple of reviews that came out in the late 80s and early 90s by Stiling and Cronin and Strong, where they looked at a bunch of these studies that actually did this. And what they found was that actually these cases, like the Varley case, are not all that common. The ability to detect it increases with more and more duration, with a longer and longer duration of a study. So suggesting that it's hard to detect, and only really long data sets allow you to actually do that, at least with these observational data sets. And this other one by Cronin and Strong actually showed that, yeah, in fact, it's not very common. And in fact, the cases that are more common are these cases of inverse density dependence, where the control factors are actually getting weaker as densities increase. And clearly, there's some critical problems with trying to detect density dependence. Having long enough data sets is really important. This was a point made by Ilka Hansky. A different school of thought emerged roughly at the same time as the ideas of the density-dependent functional regulation by Varley and Nicholson and others. And that was promoted by Andrew Wartha and Birch. And they actually said that density dependence is actually not that important for regulation of populations. And the reason for that is that populations never really get up to these levels where competition or predation is really all that important. And that's because there are a series of abiotic factors, constraints, disturbances that constantly knock the populations back, so they never get to the levels where resources are limiting or predators can actually be shown to have a negative impact on them. And they really stressed that environmental variability was key, and there was no reason why we should expect there to be these beautiful, stable equilibria. And it's really most of the population fluctuations are just driven by abiotic factors. They came to these conclusions based on the systems that they were studying. They worked on thrips, which are these little plant-feeding insects that they were studying. And what they found is that they could explain the populations of these thrips really well just by a simple measure of some key abiotic factors, like their degree day accumulation. You should know about this already. How much rainfall was present, and something about the daily temperature size, and obviously something about the previous year's population. And they could model, this black line here is the model of their prediction of what population size would be given these factors, and the little dots are the actual observations of them. And so they thought, it's really abiotic factors that set the population. The population kind of tries to grow, it grows exponentially, and then it gets knocked back or it gets throttled back by a certain amount that's due to these abiotic factors. And this was kind of an alternative competing hypothesis to go with the functional hypothesis. And as you can imagine, with any kind of debates that are diametrically opposed as to what the driving factors are, the answer probably lies somewhere in between. Of course, abiotic factors are going to be important. We've learned that insects have particular niches that are important to them, but we also know that they don't occur everywhere, and populations don't often explode. And predators can be important at bringing those back. Some of those examples of biological control that we talked about. And so there was really a reconciliation to this debate, which actually was kind of rancorous, and folks kind of got mad at each other and said nasty things at meetings and so on. And the reconciliation is basically that both things are actually happening, and that there is a region of population fluctuations that are kind of driven by stochastic effects, weather, temperature, precipitation, disturbances, things that knock the population back in this density-independent way. And that once in a while, as the population actually does get big enough, there are negative feedbacks by these density-dependent factors. There is suppression here. Sometimes this is called stochastic boundedness. That is, populations bounce around stochastically, and then when they hit these boundaries, there are factors that bring them back. The opposite actually happens here, where once they hit a floor, the density actually gets to the point where they can't find mates anymore, and the population actually goes extinct. So staying away from that floor is actually really important. That effect of kind of density-dependent mating and reproduction is often referred to as the Ali effect. So just to recap this particular section, regulation in the strict sense of the word can only really happen by density-dependent processes. But it's very hard to actually detect it in nature because of the limited nature of the data that we actually have. And yet, density-independent factors, that is, factors that can knock back populations, kill individuals in a way that is not dependent on density, like rainfall events or disturbance or things like that. Those things don't really get stronger as the population gets bigger. It's just a certain fraction of individuals are going to get knocked out when that happens. Is also important in maintaining populations relatively low. And you can have both density-independent and density-dependent factors occurring and affecting populations.